Your ears are structures for both hearing and equilibrium. So we have to try to keep those two functions a bit separate when we're studying the ear. We take a look at an ear and we can divide it anatomically into three regions. There's the external ear, which includes your auricle or pinna that collects sound, your external auditory canal, and your tympanic membrane. So those are all considered part of your external ear. The middle ear is this cavity inside your temporal bone, which houses your auditory ossicles, which are these little tiny bones. And then you have your inner ear. The inner ear is divided into three parts. You have your cochlea, which is the sensory structure for hearing, the semicircular canals, which are sensory structures for what we call dynamic equilibrium. And then you've got an area in between those two right here called the vestibule. And that's for what's known as static equilibrium. So there's really three functions. There's hearing, and then there's two forms of equilibrium: static equilibrium and dynamic equilibrium. Now you'll notice that equilibrium only utilizes these two parts that are found in the inner ear. Hearing requires parts of the external ear, middle ear, and inner ear. So what are these structures of the external ear used for? Well, the auricle or pinna collects the sound waves. Your external auditory canal, or meatus is basically a canal, directs the sound waves to the eardrum or tympanic membrane. When the sound waves hit the tympanic membrane, it vibrates. So it converts sound waves into vibrations. The vibrations then have to be transmitted through the middle ear using the auditory ossicles in order to produce fluid waves that travel through the cochlea and are detected. And these waves are determined by the different pitches of sound. So we're not going to get into extreme details in how the cochlea works, but we want to make sure we know that the cochlea is the sensory structure for hearing. Then we're going to talk about equilibrium, and we're going to differentiate into static equilibrium, which is going to use this structure right here called the vestibule, and then dynamic equilibrium, which are going to use these structures here called semicircular canals. Okay, so when sound waves hit the tympanic membrane, it's going to vibrate. The ossicles are attached, specifically the malleus is attached to the tympanic membrane, so it vibrates. When it vibrates, it transmits the vibrations to the incus and then to the stapes. So these auditory ossicles are there just to amplify and transmit the vibrations to an area called the oval window. The oval window is basically an opening to the cochlea. All right, again, cochlea is going to be the sensory structure. So malleus means hammer, incus means anvil, and then stapes means stirrup. Those are the three auditory ossicles, and that's the order you should study them, because that's the order in which they transmit the vibrations. Malleus, incus, stapes. We also have to make sure that the pressure on each side of that tympanic membrane is equal. So the pressure on this side of the membrane is atmospheric pressure. So whatever pressure the atmosphere is, that's the pressure right here. This cavity needs to be able to open up to the atmosphere. So there's a tube, all right? It's called the eustachian tube or auditory tube. Don't confuse it with the auditory canal. So here's the canal, here's the tube, all right, shown right here and right here. This tube opens up to your throat. Your throat is continuous with the atmosphere. So if you're changing, let's say, altitudes, and the pressure in the atmosphere is changing, you have to open up this middle ear to the atmosphere by opening up the eustachian tubes. And typically that's when you pop your ears, right? Popping your ears is when the eustachian tube opens and we equilibrate or equalize the pressure on each side of that membrane. If the pressures are not equal, the membrane doesn't vibrate properly. And if it doesn't vibrate properly, that affects your hearing. All right? Okay, so we just transmitted the vibrations through the ossicles. Now we want to detect using your cochlea. So the cochlea is for hearing. And then these are the semicircular canals that are going to be used for one form of equilibrium called dynamic equilibrium. And then it's kind of hard to identify, but we'll talk about this area here, which is known as the vestibule, and it is involved in static equilibrium. Okay, so where are we? This kind of helps you understand some of the physiology. Again, we're not going into extreme details. We don't have the time. But I do want you to remember that there's some conversions that are made, right? Sound waves are converted into vibrations by the tympanic membrane. Those vibrations get transmitted from malleus to incus to stapes. The stapes is sitting in the oval window. So if this stapes is vibrating, imagine what that would do to this fluid inside the cochlea. It would cause waves, all right? It's kind of like if you were to shake your hand in a swimming pool. If you shake your hand and slap a swimming pool, the water, you would make waves. And then those waves are going to travel within the cochlea, and they're going to stimulate receptor cells. The receptor cells are for hearing. That gets transmitted to your auditory area. You use your association auditory area, and you process that as sound. All right. Notice that if the waves kept traveling, you don't want them to bounce off and travel back. So you have to absorb those waves using what's called the round window. That would be if you were producing waves in a pool, you don't want the waves to reflect off the end of the pool and come back across. That would cause a lot of issues if it happened in the cochlea. All right, so that's your basic idea. Again, I don't want to go crazy because we don't have the time, but we should understand how the external ear is involved, how the middle ear is involved, and what the cochlea is for. Now, within the cochlea, there's a structure called the spiral organ, or the organ of Corti. That's this area in here that actually contains the sensory neurons. So it's the sensory neuron structure of the cochlea. All right. Okay, that's hearing. Let's talk about equilibrium. There's two forms of equilibrium. One is known as static. One is known as dynamic. It kind of suggests something. Static means not a whole lot of movement, whereas dynamic kind of sounds like you're going to have a much more dramatic movement of the head. So it's movement of the head that needs to be detected to help us maintain equilibrium or balance. So the first thing we're going to talk about is static equilibrium. It is position of the body, specifically the head, relative to gravity. All right. We also refer to this as linear acceleration of the head. All right. So if you're moving your head up and down, gravity changes, right? Gravity doesn't change, but the position of your head changes. In fact, and then affects how much force is on your head. So we need to be able to detect this. So what's utilized is the vestibule. So that's this area right here in between the cochlea and the semicircular canals. Inside the vestibule are two sacs. One's called the utricle and one's called the saccule. Inside those sacs are what's called otolithic membranes. Otolithic means ear stones. And they're actually little calcium carbonate crystals, all right, in the form of a membrane. Embedded in that membrane are little sensory neurons. Okay, so these little blue guys here are sensory neurons. You'll notice here they call them hair cells. What they have are these little structures called stereocilia. They kind of look like hairs. They're embedded in the otolithic membrane. When gravity changes, so the key to static equilibrium is gravity. You see the word gravity, I can almost guarantee the answer is static equilibrium. When gravity changes because you tilted your head down, or maybe you moved up or down, that's going to shift the otolithic membrane. Notice when the otolithic membrane shifts, it shifts the little stereocilia, and that activates the neurons. This information then goes to your brain. Your brain processes this as a change in gravity, and if gravity changes, we need to respond and maintain balance. Okay? So that's the gist of it. Static equilibrium is relative to gravity. It's monitored in the utricle and saccule that's within the vestibule of the inner ear, and it utilizes the otolithic membrane or ear stones or ear crystals. So then what is dynamic equilibrium? Dynamic equilibrium is more rotational. All right, so when you're rotating your head side to side, gravity is not changing, but we need to be able to monitor that shifting of the head so we don't lose our balance. This is known as dynamic equilibrium. So we call it rotational acceleration deceleration. The structure's housed in the semicircular canals. So those are these loops, right? So the semicircular canals are utilized to monitor this dynamic equilibrium. Inside the semicircular canals, there's a little structure called the cupula, which is found in an area that's a little bit bigger. So it's hard to tell, but there's an area in the semicircular canal that's a little bigger, this little area here, called the crista ampullaris. In there, there's this little cupula, and the sensory neurons are embedded in there, kind of like in the previous slide. But now when you shift your head right to left, the material, there's fluid in here that shifts. And when the fluid shifts, notice when it shifts this way, it moves that cupula, which stimulates the neurons. So now your body is monitoring a dramatic or dynamic movement of the head right and left. And just by how the liquid is shifting in these semicircular canals is how we stimulate the neurons. This goes to your brain. Your brain processes this as a major change in head position, and therefore we need to balance. All right, so the two equilibriums are similar, but you want to make sure you keep the structures separate. All right, so dynamic equilibrium utilizes the semicircular canals. It utilizes the cupula, which is found in the crista ampullaris. And dynamic equilibrium is when your head is rotating to the right and left. This is a chart I came up with years ago to try to help. I don't know if it helps, but it kind of separates the three functions out into kind of a diagram. So you'll notice that hearing is going to utilize your outer ear. Talked about those three parts. It's going to utilize your middle ear. We talked about the eustachian tube equalizing pressure, and we talked about the auditory ossicles and their order. We talked about the fact that the stapes is going to vibrate in the oval window, and when that occurs, it's going to produce waves of fluid known as perilymph. That's just the liquid in the cochlea. All right, and that liquid is going to then activate the spiral organ of Corti, which contains the receptors. And that spiral organ of Corti is housed inside the cochlea. All right. We're not going to talk too much about this cochlear duct and endolymph. It's just that there's a couple different fluids in that cochlea, but ultimately it's the waves of fluid that get sensed by the spiral organ of Corti that contains your receptors. So all this stuff in black text is for hearing. In purple text is for static. So remember, it's the position of the head in gravity. The structures are housed in the vestibule. They're known as the utricle and saccule. The vestibule is often referred to as egg-shaped, and that in that egg-shaped vestibule are these two sacs. These sacs then have the otolithic membrane. All right, we're not going to talk about the macula of the vestibule, but shifting of the otolithic membrane is what is detected as a change in gravity. So all the things in purple, think of static. Dynamic equilibrium, rotational, acceleration, deceleration, monitored in the semicircular canals within the crista ampullaris, and it utilizes the cupula, which is found in fluid also known as endolymph. So I don't know if this helps. Again, to me, color coding helps because everything in black you want to associate with hearing. Everything in purple you want to associate with static equilibrium. Everything in blue you want to associate with dynamic equilibrium. So that brings us to the end of this chapter. Good luck. Let me know if there's anything I can do to help. Keep in mind the best thing to help you study are the animations that I share. It's really hard to understand how these things work when you don't see them working. So watch those animations until you start to pick up how these things are working together. Good luck.